The chemical digestion. In this module, you will learn about the chemical digestion that takes place in the human body. Digestion involves two processes, physical and chemical. Physical digestion breaks food into smaller pieces, whereas chemical digestion refers to the breakdown of complex forms of proteins, carbohydrates and fats into simpler forms so that they diffuse into the cells. Chemical digestion is carried out by various enzymes produced in the body. The different parts of the alimentary canal secrete specific enzymes. Before the process of chemical digestion can be understood, it is important to know that carbohydrates and proteins are made up of chains of polymers which, when acted upon by enzymes, are converted into monomers. Fats are not polymers, but molecules of fatty acids linked to glycerol. Enzymes will separate the fatty acids from glycerol. Let us first look at the chemical digestion which begins in the mouth. Food in the mouth stimulates the salivary glands to secrete saliva which is mixed with the food. The mucin in saliva helps to soften the food, while chewing helps to break the food into smaller pieces. Saliva contains salivary amylase, the enzyme which digests cooked starch to maltose. The food is rolled by the tongue into small, slippery, spherical masses called bolus. These are swallowed and enter the esophagus via the pharynx. Proteins and fats remain intact at this stage. Renin from prorenin coagulates the soluble protein in milk called casinogen into its insoluble form casein. Now it can be hydrolyzed by pepsin. The pepsin acts on proteins and converts them into polypeptides and peptones. On entering the small intestine, the chyme stimulates the intestinal gland to secrete intestinal juices. It also stimulates the pancreas to secrete pancreatic juice and the gallbladder to release bile. Both the pancreatic juice and the bile moves into the duodenum through ducts. The enzymes in the pancreatic juice are the protein-digesting trypsin, the starch-digesting amylase, and the fat-digesting lipase. Trypsin, initially in its inactive form trypsinogen, is activated by an enzyme called enterokinase from the intestine. Trypsin hydrolyzes proteins and polypeptides into peptides. The intestinal juice also contains erepsin or peptidase, maltase, sucrase or invertase, lactase, and intestinal lipase. The intestinal juice is also known as succus entericus. In the duodenum, the bile salts from the liver emulsify fats into smaller particles suspended in water. This increases the surface area over which the lipases can act. Lipases convert the emulsified fats into fatty acids and glycerol. As we know, the starch is digested in the mouth itself. The remaining carbohydrates, including the leftover starch, are digested only in the small intestine and not in the stomach. The remaining starch is converted by pancreatic amylase to maltose. Maltose is simplified to glucose by maltase, lactose to glucose and galactose by lactase. and sucrose to glucose and fructose by sucrase. Thus, the end products of carbohydrate digestion are simple sugars. The cellulose is not digested in the alimentary canal. The digestion of protein begins in the stomach, where some part of it is digested. The undigested proteins which enter the small intestine are converted by trypsin to polypeptides. The polypeptides produced are further digested to amino acids by the enzyme erepsin in the intestinal juice. The end products of protein digestion are thus amino acids. Thus, you have studied the complex carbohydrates are converted into monosaccharide, proteins into amino acids, and fats into fatty acid and glycerol, which are easily absorbed by the cells. Let's summarize. Digestion involves two processes, physical and chemical. Chemical digestion refers to the process of converting complex forms of carbohydrates, proteins, and fats to simpler forms so that they can be easily absorbed into the cells. The cells in the stomach produce hydrochloric acid, pepsinogen, and prorenin, which are present in gastric juices. Trypsin, amylase, and lipase are the enzymes found in pancreatic juice. The intestinal juice also contains erepsin, maltase, sucrase, lactase, and intestinal lipase. The digestion of carbohydrate begins in the mouth. The mucin in the saliva softens the food, and the salivary amylase converts starch into maltose. The end products of carbohydrate digestion are simple sugars. The digestion of protein begins in the stomach and ends in the small intestine. The end products of protein digestion are amino acids.